right, I think it's 12:01, Trace. Might as well get going. Lots of good stuff coming from UNH, so probably want to get going. I will defer to our committee chair, research committee chair, Tracy from Brown and Caldwell, and she'll take it from here, like she does every month for our meetings.
Great, thank you so much, Janine. So this is, something new that we're trying. So we've asked Dr. Paula Mouser to be our first researcher to come in and provide us with what she's doing, some of the research she's working on. But that's really what this committee is about, is pairing researchers with the industry so we can really learn what we need to learn to be better. So with that, I'm going to introduce Dr. Paula Mouser. She's an associate professor of civil environmental engineering at the University of New Hampshire, the administrator for UNH's Bachelor's in Environmental Engineering undergraduate program, and the director of UNH's Wastewater COVID-19 monitoring program. She'll talk a little bit about that. And her research applies genomic tools to understand microbiological communities and pathogens in engineered and natural systems. She also utilizes high-resolution analytical chemistry to study the fate of emerging contaminants, including the PFAS, and polyfluoroalkyl substances, pharmaceuticals, and personal care products in engineered freshwater and estuarine systems. She has co-authored more than 50 papers and collaborates broadly across disciplines and stakeholders. So thank you so much for presenting what you're doing. And she's asked that if you can hold questions to the end, you can put them in the chat and we'll get them answered at the end.
Oh, thank you so much, Tracy and Janine, for the invitation to present. I didn't realize this was the first committee meeting we were doing this at, so it's my pleasure to step us out in a UNH direction. Go Wildcats! I've prepared some slides that are really research examples on PFAS and COVID wastewater studies that we've been working on for the last 4 years. And I don't have a lot of background because I know this is a very educated group. And so I'm not going through what PFAS are or SARS-CoV-2 virus. I'm just going to jump into some research examples. So I'll share my screen right now. And then you can tell me if this is coming across okay. Does that look okay? All right, wonderful. Yes. All right, great. So I see some familiar faces in the audience, and I know that there's going to be some really hard questions come my way at the end of this talk. But I know that you all know that wastewater facilities play a central role in collecting, concentrating, and also the way that we're talking about it in SARS-CoV-2 and PFAS, fractionating some of these things into the solids and the liquid phases as they move out of the facility. Sorry, I'm
going to interrupt 1 second. Is everyone else seeing your screen, the slides? I'm seeing build forward with confidence, or is that just me?
That's you. I
okay.
See.
It's just a me problem. So I will try to figure this out. Thank you. Sorry for interrupting.
Someone will put in the chat if they're having that issue, please.
Should I keep going? All right. So I'm broken up my talk to start off on some examples of PFAS, first in the New Hampshire Seacoast area, and then working on some data mining of the Vermont and New Hampshire databases that are available with PFAS information. And then I'll touch on some of the SARS-CoV-2 wastewater surveillance that we've been doing and some initiatives that the state agencies have kicking off right now with respect to COVID monitoring and wastewater. So I don't do this work in isolation. I have a lot of great colleagues that I work with to understand some of the topics that I'm talking about, including my colleague Jim Malley here in the department, as well as Jenna Luke, who was a former postdoc, and several students. Many of them have gone on to work in some of the firms in the area. So the work that the students are doing usually have their image up at the top, so you know who's responsible for the great work. And it's usually not me, it's the students. So we have a lot of great collaborations going on, funded through Sea Grant funding, as well as other agencies. So one of the questions we started asking about 4 years ago was, if we looked in the New Hampshire seacoast area, are there differences in influent, effluent, and sludge PFAS concentrations and compositions? And so we're looking at, what's coming into, what's going out of the facilities in the aqueous and the solid phase with some of these studies. And one of our first studies looked at 6 wastewater facilities that were located in tributaries of the Great Bay here on the seacoast, as well as 5 locations in the Great Bay itself. And Ellie did a couple of sampling rounds where she went out and she sampled influent and effluent and analyzed these for 24 PFAS constituents. And then we went back and we looked at the different unit processes within the facilities to see how things changed during treatment, to figure out what the fate of those compounds were in different types of treatment designs. And one of the things that we saw pretty quickly was that most of the facilities had pretty similar compositions of PFAS coming into and out of their systems. And so if you look across the left-hand side, these are the 24 constituents we looked at. And you see that in general, across the bars, most of them have the same carboxylic acids, many of the same sulfonic acids, as well as a few precursors that are in the influent and effluent of these facilities. What jumped out at us was that commonly the influent concentration ended up being lower than the effluent concentration. And this was really perplexing, and we see this in the color change. Gray means detected, red means that the effluent, we saw higher concentrations. And so we're really wondering what would cause us to see higher concentrations coming out of the facilities, or other facilities seeing this. So we wanted to dig in a little bit more to that question with some of our work. And when we looked at the different unit processes, what we saw was that after the wastewater moves through secondary treatment, we see that there's a lot more PFAS that's liberated, a dramatic increase in the PFAS after secondary, which tells us there's something going on during secondary treatment that's causing either liberation from solids of some of these constituents or some sort of partial breakdown, generating compounds that we can then measure. And this was really fascinating to us. So we began to look into, are there biological processes that can transform these PFAS compounds? And generally, there are not microorganisms that can break these down fully, mineralize them to fluorine or carbon dioxide. But there are partial transformations that can occur to certain of these compounds that can really affect how we see them when we try to measure a subset of them. One of the examples that we think is relevant to wastewater relates to these fluorotelomere alcohols. Microorganisms can really easily modify the alcohol end of these compounds. They create carboxylic acids through some sort of metabolic or co-metabolic process. Those compounds generally go undetected because they're not compounds that we're really looking for. But then those can break down further to other compounds when the very end of that is cleaved potentially or modified one more step. So a fluorotelomere alcohol could potentially create something like PFOA, and there's other combinations of compounds that can generate some of the common short chain carboxylic acids and sulfonic acids that we see coming out of these facilities. So this is actually really important in the secondary treatment process, where we have lots of microorganisms, lots of activity going on, lots of enzymes that are promiscuous and maybe cleaving compounds like this that they're not necessarily designed to cleave. And we know that this is happening because we have a new test that we're applying that's called the TOP assay. Many of you have probably heard of it. It's a total oxidizable precursor assay. It uses a chemical approach to basically oxidize, really harshly chemically oxidized those PFAS constituents into more terminal products, and many of those are compounds that we can measure. So one of the things that Ellie did was she looked at how much oxidizable precursors were present in the wastewater in these facilities. In March, in the influent and effluent, as well as in July, influent and effluent. And what she found is that in March, there's a lot of oxidizable precursors in the wastewater, whereas in July, there's a lot less. And generally, there's a lot more oxidizable precursors in the influent than there is in the effluent. And what that tells us is that during warm months, there's a lot more oxidation going on in the sewer shed before it gets to the facility, as well as in the facility, that basically is transforming those compounds, those partially degradable compounds, into some of the terminal metabolites that we're seeing. And this helps explain why we might see a lot of increase in the wastewater treatment facility in the effluent, especially during the warm months, which is what we saw commonly. I think that's really important for implications for monitoring as well as for regulation, because if we have changes in the way that these compounds are degrading, increases in the summertime could have an effect on, on what the concentration and loads are coming out of these facilities. So we're also really interested in how the aqueous phase differs from sludges. And so we did include sludge analysis in the studies that Ellie was looking at. And we see really dramatic differences in the relative composition of these compounds, where the influent and effluent are primarily comprised of a lot of the carboxylic acids and sulfonic acids that are small, whereas the sludges, as shown in the dark brown and the black, have primarily the long chain compounds that are sulfonic as well as precursors. And so we know that there's some partitioning that's going on in the facility that's causing some of these compounds to drop out. This is also important when we think about the recycling of the sludge back in that secondary treatment biological container. For recycling sludge with high amounts of these long chain compounds, potentially fluorotelomeres, they have more time to react in that basin and break down partially. So we started looking at these compounds in terms of short chain and long chain. And what we saw was, as we moved through the facility, we see fractionation of these compounds. The shorter chain compounds like to go into the aqueous phase and out the discharge. And we can see that because the lighter orange gets more abundant as we move through the facility, whereas the sludge becomes enriched in the longer chain compounds as well as those precursors. So there's this natural fractionation of these PFAS into the phases where they like to be based on their affinity for solids versus the liquid phase. So this is also really important because we're sending out compounds that are potentially more mobile, that like water more, and we're fractionating into the solids, the ones that like to remain in that solid phase or that are larger. And I think this has really important implications for biosolids handling. In addition, not only do we see this fractionation that occurs, but we also see the concentrations are significantly higher in the solid fraction. So when we look at this total sum of what we're detecting in the solids, in general at these facilities, the concentrations are about 3 orders of magnitude higher in the solids than in the aqueous phase. And so not only are we fractionating, but we're also concentrating in the solids when we are handling these two different phases. So we used our information to try to estimate the log KD values in these facilities, and these are comparable to what we're seeing at in other studies. And naturally, the shorter chain compounds have lower log KDs, which means that they prefer to be more in the aqueous phase, which makes sense. That's what we're seeing with our data. And then as those compounds get longer in their chain length, they would prefer to be in that solid phase. In addition, we see that at the same chain length, the sulfonic compounds, PFOS, are 10 times higher in the solids than the PFOA is in the sludge. And so not only is it a chain length, but it's also a functional compound that's that's related to how much affinity those compounds have for the solids versus the aqueous phase. And there's a really big break between that 6 and 8 chain length for where those compounds like to remain in the solids versus the aqueous phase. So during the pandemic, the students were not doing a lot of field work, and so one of the things that we focused our time on was some of the data analysis of these large databases that are being amassed by state agencies. So lots of samples have been collected by many of you out there, and they're being deposited in these databases. And we felt it would be a good time for us to start looking at some of these data and asking really specific questions about, you know, what's happening in these facilities and across the states. And so we ask another question, you know, do the sources that are discharging to facilities or the sludge handling processes influence the signature of the PFAS that we're seeing? And the databases that we've been looking at include a really nice report conducted by Weston and Sampson in collaboration with Vermont DEC that looks at PFAS in wastewater treatment facilities and landfill leachate, as well as databases from Vermont DEC and New Hampshire DES that have both biosolids and wastewater information. And these have been a great resource for my students. So we started applying what I would call fingerprinting approaches to try to back out whether different sources relate to the signatures that we're seeing in the facility. And one of the approaches that we use is called non-metric dimensional scaling. It's a multivariate statistical approach that allows you to project lots of information on a couple different dimensions. And we worked with the Vermont DEC to basically categorize facilities based on whether they had industrial discharge, residential or commercial or landfill leachate dischargers. And what we saw was that the landfill leachate accepting influence had a really distinct signature that was different from other industrial and commercial and residential primarily accepting facilities. And we see that because the yellows are clustering together in this two-dimensional breakdown. And we can look a little bit further and project this in a Venn diagram. And what we see is that the landfill leachate was primarily associated with the compounds that are listed in the left-hand side, whereas there weren't a lot of individual PFAS that were solely associated with industrial residential accepting facilities. This is pretty helpful for us too, because if we see a certain signature of compounds, it tells us a little bit about what the discharges are. And it also tells us that if we're seeing them in the influent, we're also probably going to see them in the effluent and possibly the sludges too. And so that helps us get a handle on you know, where some of these signatures might be coming from. We also we looked also at whether or not there were differences in biosolids handling in terms of PFAS. And we did this by classifying again into short chain, long chain, and biosolids. I'm sorry, short chain, long chain, and precursor fluorotelomers. And what struck us about this analysis is that there didn't seem to be much different between the biosolids that were not processed or lime stabilized handling, but there were really very big differences in the PFAS composition of those biosolids that were anaerobically digested versus those that were composted. The anaerobic digested samples had a lot more of the precursor fluorotelomer and longer chain compounds, which makes a lot of sense because you are doing an anaerobic digestion, and there's a lot of breakdown of compounds as well as gases that are coming off during that process that might liberate some of those smaller compounds. Composting on the other side had a lot more of the short chain compounds. And this is something that we couldn't explain very well. It could be related to the agents that are added or the way that the solids are degraded anaerobically. So we're really fascinated by why we would see big differences in the different handling. Also really interested in what this means in terms of mobilization if these biosolids are used or are field applied. We applied the similar fingerprinting approach to the same data set to try to get a handle on whether or not there was a significant effect of stabilization on the PFAS signature. And we did see that there was this really distinct breaking out of anaerobically digested versus composted type samples. And when we looked more specifically, we saw that composting had this really distinct signature of a lot of these short chain carboxylic acids, as opposed to the lime stabilized anaerobic digested or the other solids that were not necessarily processed. So that was also really fascinating to us to know that primarily some of these compounds were associated with certain handling. We're really interested in adding to this database to try to flesh this out a little bit further. We didn't have a lot of samples to use in this analysis, so I think there's a lot of research that could be done here to better understand how these solids handling approaches relate to the signatures that we're seeing and what those signatures mean in terms of mobilization. So a lot of great work being done by the students. The last project we're working on with PFAS right now is with the Vermont DEC. We're looking upstream. We're trying to take a P2 approach to identify the low-hanging fruit for industries or commercial entities that we might be able to target for reduction of PFAS before they come in the facility. And so here's another example of how we're using the traditional approach for analysis of PFAS, as well as this TOP assay. The TOP assay is the total oxidizable precursor. And so what you're going to see in each of these sample locations in this kind of network of a sewer shed, which is above the wastewater facility, is that there's two pies. One of them is the top assay, tells us how much oxidizable precursors there are that generate metabolites. And then always the smaller pie is the PFAS compounds and composition that we get through the more traditional analysis that we're doing, the isotope dilution method. So what we see is that when we don't use the top assay, we miss a lot of information. We miss a lot of the mass. There's a lot of mass in the sewer shed that we're not actually seeing because we're not targeting all of the compounds that are there. And many of these compounds are degradable. And so in some cases, the concentrations that are there that we don't see are 10 times or more higher than what we're seeing with the isotope dilution methods that's generally used. We can also get a handle on which locations we're seeing a lot more mass. And surprisingly, we saw that there's a lot of PFAS coming out of residential areas. And when you think about this, it kind of makes sense, especially in a state like Vermont that doesn't have a lot of industry, that the residential areas where we have a lot of this kind of legacy PFAS, things that are coming through, they might be small mom and pop commercial businesses that aren't very well regulated. So I think there's a lot to be learned about these studies that go up into sewer shed and selectively target certain locations for sampling to try to get a handle on, where is the mass coming from so that we can eliminate that or do a better job of reducing it at the source so that we don't have to manage it in the facilities. So I know that's a lot of information and I'm happy to answer some questions when I wrap up a couple more slides on COVID, but just kind of an example of some of the different projects that we have going on. Most of these are master's students and some of them involve, field sampling. Many of them involve data mining to try to understand, what's happening with PFAS fate in wastewater. So I have about 5 more minutes, I think. Does that sound right? And I was just going to give some examples of about 5 minutes.
Yeah.
Of SARS-CoV-2 monitoring that we're doing here on campus and also in some other areas across the state. And so this is also a really big team. We have 8 students that are participating in terms of our sampling here on campus. And then I've great staff and students that are involved in some research studies. And we've been monitoring at the campus for almost 2 years now. We've also been looking at what's happening in our wastewater treatment facility. In addition, I have a student looking at the fate of SARS-CoV-2 in wastewater facilities here on the seacoast. So I'll talk a little bit about that. Durham is using wastewater surveillance as information for making decisions about health here. And we have an extensive network of sample locations. We have 26 residence groups, and they range from 1 to 4 halls each. We're deploying gauze swabs in the manholes for a period of 2 to 3 days at a time. And those swabs are stacked, they're deployed, and then they're retrieved. And we process them here in my lab. We monitor about 21,000 students, staff, faculty, and community members with this approach on and off campus. And we collect samples 3 times a week. And in the lab, we use some really cool biotechnology tools. We target primers similar to what you would get targeted if you were doing a nasal swab testing in N1 and N2 genes, as well as a human waste gene. We use a magnetic hydrogel to concentrate the virus out of wastewater and clean it up. And then we use an automatic extraction technology along with some microfluidic techniques to quantify the number of biomarkers in our sample. And this is highly sensitive, and it allows us to track things on a per 100 ml basis. And it's been working very well to tell us what's happening across campus. Here's an example of what our dashboard looks like. So the decision makers at UNH have access to this information. They know where COVID is being detected in the residence hall group at any time. We've also overlaid this information with individual test data that comes from the nasal swab testing. And so we can get a handle on whether or not our detections align with known infections. And if they don't, then we are expecting them to come soon, or we know that we're missing infections that are happening in certain areas. So I report out a couple times a week to different COVID response teams to make decisions about, how we should move forward with some of the infection spread that's happening on campus. And just to give you an idea of how truthful the wastewater data is related to absolutely known infections in our residences, the blue are wastewater quantifications. And so they're in copies of our biomarker per 100 mils of wastewater. And the stars are known infections. So those are positivity. So when they go up, we have more individuals in that residence hall that are infected. And what you probably see here is that almost every time, if not every time we see a star, we also have a detect in the wastewater. Our approach is really highly sensitive. And when we have higher positivity, we also have a higher signal. And so we're very confident that the wastewater corresponds exactly to known cases. And as that network builds, we see increased sensitivity at the location near the monitoring point. So residence halls that are closer to the monitoring point are going to shed a higher, amount of that virus that we can detect better closer to that monitoring point. And so things build as you go down that network. And it's been really fascinating to see that we can predict what's happening in the residence halls about 1 to 2 days before we hear reports from Health and Wellness about infections in those halls. We can also detect vaccinated and unvaccinated individuals, which makes it a really useful tool for knowing what's happening with our campus. And we've had some really cool student projects related to wastewater monitoring. One of them was a capstone project that just finished up. We had some help from Woodard and Curran as the sponsor. These students used a camera system and tracers to try to get a handle on how things are moving through our watershed. And they deployed this into our monitoring points for a certain amount of time. They added tracers in the buildings. And then they mapped out how those tracers were recorded in the camera system through time. And just to give you an indication, when we have a small residence hall area, we see a lot of spread of that tracer in our monitoring point, which means it's going to contact our swab readily. We're going to have a really good interaction of that signal with our swab. Every time we added a tracer, which are the peaks on these plots, we see that it takes a long time to push through that monitoring point. On the right-hand side, Stoke Hall is a very large hall, 800 students, and that tracer moves through really quickly through the system, almost in a plug or a slug flow. And so it doesn't interact with our swab as much, and if we don't sample properly, we miss that slug. So So the way that the tracer, which we're using as a surrogate to the virus, moves through the system has implications for how we need to monitor it. And so we're trying to use this information to improve our wastewater surveillance program moving forward. So I have a few more I have a few more slides on the fate of wastewater, but I think I'll just hold them for another time since we're about out of time. Does that sound good?
Yeah, maybe they'll come up in the questions that we've already started to come in.
Sure. I'll just go to the last slide then. And I'll just say some of you may have received some information from the state regarding a wastewater surveillance program that we're kicking off with New Hampshire Public Health Services, DEA and DES. There's a webinar coming out on that soon, and I'm happy to answer questions. And if there are other utilities or communities interested in PFAS related projects, I'd be happy to talk to you about them. Also wanted to mention that Stuart Grandy, my colleague, and I have ordered and are receiving an instrument to be able to measure PFAS in soils and biosolids, and it should arrive in the next few weeks. So we'll be focused this summer on methods to be able to get that up and running. So,
oh, outstanding. Since Tracy had the first question, Tracy, I'll take over the Q&A functions here.
All right.
Dr. Mouser, Tracy was asking, did you look at different types of composting? And if there was a difference between the composting technologies type? So like in vessel versus other, static pile or something like that.
Well, that's a great question, Tracy. There weren't enough samples to do this. I think in total there were 5 or 6 composting samples in the data set, but obviously there's a great need to be able to trace the samples, the source of the samples through that processing, and then see how the PFAS change as a result of the handling. And so I'm hopeful that there are studies that are going to be done like that if they're not already underway.
I'm keeping a little list of future research ideas, so thank you. That's definitely one. And then Jason, one of our New Hampshire members, had a couple of questions. The first was, were there seasonality differences observed in sludge concentrations or signatures similar to the influent and effluent?
Oh, that's a good question. We only sampled the sludges during one of the sampling events. We didn't look over season. I would guess, Jason, and I knew he was going to ask me the hard question. I would guess that that there probably are also differences in how they're accumulating in the sludge. Temperature as well as pH are likely to influence, how those are moving between phases. And so yeah, I think that's a real open question.
That was new information to me too, the seasonality of it, and certainly important for regulations, I would say. And then his other question was, would a multi-factor treatment or handling approach such as lime stabilization and then compost benefit systems? Because it looked to him like they were doing the opposite. One was creating long chain, one was creating short chain. So some type of a multi-stage process. Maybe.
Oh, it's interesting you asked that. I'm working with a student right now, Jessica Nekovich, who's doing a study looking at biosolids handling, and she's put together a really interesting kind of closed loop handling approach, and I wonder if thinking through how we could actually process these solids in a couple different ways, to minimize the need for land application or other types of handling, if that would also have a dramatic effect on PFAS, especially if they involve both really anaerobic and really aerobic processes.
That will be interesting. Great questions, Jason. Also, here's a question from Ned. Doesn't the Vermont data suggest the largest source of PFAS at the Essex Junction is the residences? And
right.
Those PFAS are likely coming from household products, and yeah, this is
yes, Ned, you hit it on the head with that, and so from a regulatory standpoint, it's not very helpful, is it? Because it's going to be really hard to go into those residences and ask people to stop using certain products or try to work with, I don't know, commercial commercial agents to try to reduce the use of PFAS because they're just everywhere. I think it creates a real regulatory nightmare if that's really what we're seeing, especially when we don't have a lot of industry in some of these small communities. The low-hanging fruit is really hard to get at. Yeah.
Great. So that is it for open questions. Folks can also raise your hand. That's an option down at the bottom if you want, because I can actually let you speak, I think. But since there are no other questions, I'm going to kick it back to Tracy, our chair.
Sure. And if you do think of something while I am talking, please, put in the chat, raise your hand. We want to get your questions answered. But no, thank you, Dr. Mouser. I greatly appreciate this. And this is very fascinating information. And, as things progress, we'll definitely would like to have you back to provide updates of how the research develops.
Thank you so much. It's been my pleasure.
So it looks like, do we have a question? Okay, Ned has another one. Is it correct to say that the total mass of PFAS is staying the same throughout? The difference is only in which are showing up in the analyses.
Oh, no, that's another really good question. Absolutely, I think you're right. We're only looking at, in most of these studies, there was only 24 constituents that were targeted. The top assay liberates some into those 24 compounds. And so if we were to be able to look at all 5,000, we might be able to do a really nice mass balance, but obviously that's just not possible. I think we're getting closer with some of the expanded standard methods that the EPA is coming out with, but we have a long ways to go to close the mass balance loop. So it looks like we're generating PFAS, but in reality, what's happening is that there's probably just a lot more coming in than we're measuring. And some of it's being partially degraded into compounds that we can see. Yeah.
Great, thank you. All right. So if there's nothing else, and as I said, please ask if you think of something. So we are the research committee for NEBRA. And these are the type of things that we like to bring to our members and to you all, is working with researchers to answer these questions. And, Janine was saying, I'm writing that idea down. We do have a survey out there that Janine has sent out. So we're asking you all, our members, for your research ideas. Where do we want to move forward? So we try to pair researchers with others in industry, or like we did with Jessica last summer. She was an intern for NEBRA looking at the phosphorus data with Essex sorry, I cannot talk today, Essex Junction. And it's a great opportunity, and we're trying to do more of that, where we can potentially pair some potential interns with either a utility or some consulting firm, or a combination, and that would provide great experience for the students, working with people like Dr. Mouser to get some research done and get some of that knowledge transfer. So we're always looking for members, people to help. We do meet monthly for about 15 to 30 minutes. It kind of just depends. Sometimes we get away and we're so excited about new projects that we hear coming out that we just talk about it, but it's not a heavy lift. We are trying to come up with some intern research topics, some ideas of how we can structure this so we can get some internships going on, pairing the students. So whether it's, you have a topic idea, or you're willing to work with an intern or work with someone to help this research be developed, and that's share with all of our members. We really want to be open. And the other thing is, what you see is just trying to gather all this information that we're getting from our researchers and having it in a centralized location for you all to access. So like, we have a list of researchers, people we've worked with in the past, and an idea of what what they're doing. So if you have a concern or topic, you know, reach out to Janine, reach out to myself or someone on the committee, and we can help guide you to the right resource, or at least get the conversations going. And that's what we're here to do. And so we're always looking to help with that. And anyone have any questions about the research committee or any follow-ups for Dr. Mouser?
You can raise your hand if you want to
Yes.
get on the committee list. I'll be happy to add your name. But that really was a great project that we did last year with UNH student Jessica, who also ended up getting gainfully employed as a result. So that's all good.
Yep.
Viraj is asking about biosolids destruction methods available. And I'll just say that we have been tracking that. Certainly pyrolysis and gasification are at the top of the list as far as commercially viable. But supercritical water oxidation, what are some of the other destruction technologies that we're looking at?
Is it hydrothermal liquid fracs?
Yep, and we will be having some lunch and learns coming up on that. So thanks for mentioning that. We are happy to look into any technology you're interested in and try and learn more and teach you more about it. So.
And I know you all have Janine's email, but I added mine to the chat, and hopefully everyone can see that. You know, please reach out to either one of us or both. If you want to be a part of it, if you have if you need the copy of the member survey because you missed it, we'll gladly resend it out. You know, or if you have any ideas, we're here to work for you and with you to get these topics kind of figured out or started at least.
Yes, a lot of research questions still out there. All right, thanks, Jason. I got that suggestion. Appreciate it.
Okay.
Ned's just trying to connect Dr. Mouser with Linda Lee on a lot of that research as well. So that's terrific. We'll do that, Ned.
Yep. Thanks, Ned.
All right.
Thank you.
Tracy,
All right.
It's a little bit longer than normal research committee meeting, but
Yes, well worth it. So thank you so much, everyone, for joining. We appreciate the time you took out of your day to listen about PFAS and about the research committee. And thank you, Dr. Mouser, for taking your time.
Sure thing. My pleasure.
All right. Bye, everybody.
Keep up the great work.
Thanks.
Yeah.